a lot of fun. This is actually my third one, but second one in this series. We're doing a series on the methylation, and this second one we're concentrating on the vitamin D receptor, which is actually part of the methylation pathways. And to me, I consider it to be one of the most pivotal and critical parts of the pathway. It has so much effect on the rest of the body, and methylation is important in making the vitamin D receptor work, in absorbing vitamin D, and in making our adrenal hormones and so many other things that we'll learn on this webinar. So thank you so much again for hosting it. So let's just jump in, because this topic, like you say, Dr. Turi, this topic is huge. The vitamin D itself, we know, has so many beneficial effects in the body. And one of the overlooked things that one of the things that we haven't addressed is the vitamin D receptor. So in order for vitamin D to be balanced in the body, that vitamin D receptor has to work effectively. So let's dive in. Oh, let me back up, because this is kind of cool. This picture on the front there, that's the vitamin D receptor. It's a kind of blown up from an electron micrograph, and it's pretty amazing. It looks like a little party. It looks like a lot of curling ribbon going on there, so it kind of amplifies our excitement about this receptor. So in the vitamin D, you know, probably my whole audience is aware of how exciting and how interesting and how important vitamin D is in the body, and the multi-system effects of vitamin D and its metabolites in humans. No longer is vitamin D regarded simply as a steroid hormone that prevents rickets in children. Rather, its roles in epigenetics, diabetes, motor function, cell replication and proliferation, immunological processes, to name a few, have opened the door for large population-based studies, and there are a lot of studies going on all the time on the importance of vitamin D. And such studies have aimed to specifically define the role of vitamin D in these disease states and how alterations in vitamin D sufficiency affect the risk of development of such conditions. So just to give you kind of an overview and maybe some, kind of some hints about what happens in the body with vitamin D. We have 7-dihydrocholesterol, and that's in our skin. And when we're exposed to solar UVB radiation, that's converted to D3. And in our diet, we get a certain amount of D2 and D3, and that's converted to D3 in the body. And then in the liver, it's converted to the inactive form, 25-hydroxy D3. And then through the kidney, and then it's expressed into the active form, the 125-hydroxy D3, which has an effect throughout the body, from breast, colon, prostate cells, inhibits clonal proliferation. In the macrophages, it increases microbicidal activity, killing bugs, essentially, induces differentiation in immune cells. It helps improve hematopoiesis, or the production of red blood cells. In the pancreas, it increases insulin secretion, and also the receptors, insulin receptors, which we know are important for preventing insulin resistance. And in the intestine, it increases the absorption of calcium and phosphates, increases bone mineralization, osteoclast differentiation, decreases left ventricular hypertrophy in the heart, decreases parathyroid hormone synthesis and release in the parathyroid gland, and increases smooth muscle cell proliferation and reduces inflammation in the body. I think that's pretty big. We all know, some of these pieces, but when you start to look at the whole picture of what vitamin D does, it's pretty impressive. So getting our proper amounts of vitamin D in the body is also very tricky. As you can see here, getting it in your diet and getting it from the sun can be important, and a lot of us don't do that. We don't get out in the sun. So many of us are sunophobic, or we're not consuming it in the diet sufficiently. So we're taking it in supplement form, but it also has to go through the liver, the kidneys, and then ultimately the vitamin D receptor. So a lot has to go on before we get our vitamin D levels sufficient in the body. And we know that deficiency is rampant. I know a lot of you out there are looking at blood work where we're seeing a great deal of insufficiency in our blood work. So I'm seeing different values of the deficiency, and the CDC said 32% of children and adults throughout the U.S. were vitamin D deficient. The National Health and Nutrition Examination Survey found that 50% of children aged 1 to 5 years and 70% of children between the ages of 6 and 11 are deficient or insufficient in vitamin D. And Dr. Hollick did a study and estimated that 50% of the general population is at risk of vitamin D deficiency and insufficiency. But we really kind of think it's a bit higher. Life Extension Foundation conducted an analysis of 13,000 blood tests of dedicated supplement users in 2009. The startling results found 85.7% had what they considered insufficient vitamin D. And they were taking over 600 IU each day of vitamin D, but 85.7% still had 25 hydroxy vitamin D blood levels below the minimum protective threshold of 50. And those of us in natural medicine know that we like to see it even higher than that. So this number really approaches a good percentage of the majority of people are low in vitamin D. This is the current belief is the reason for that is that we have poor vitamin D absorption, and it's often a problem in people with gastrointestinal disease or where surgery has removed some of the stomach or small intestine. And most of the time people who think that they have poor absorption of vitamin D are just simply not taking enough. And that's their kind of the current line, I think, in natural medicine as well. It's like, okay, let's just crank this vitamin D level up, and we're still seeing that it's not increasing that vitamin D level in the blood. So there's still clearly something amok. And just a reminder of the different types of vitamin D here. The ergocalciferol is the vitamin D2, the cholecalciferol, vitamin D3. You're going to see these terms throughout the webinar, so I just wanted to kind of define them there. The calcidiol is the inactive form. It's also called calcifediol, and the calcitriol is the active form of the 1,25-dihydroxy vitamin D. But, so, here's the trouble with vitamin D and calcium supplements. In a surprising twist, a recent study by the North American Menopause Society found that hypercalciuria, or high urine calcium, and hypercalcemia, or high blood calcium, is commonly occur with vitamin D and calcium supplements. So a lot of people are taking these, and they're finding that this stuff is just sitting in their bloodstream. These findings may have relevance to the reported increase of kidney stones in the Women's Health Initiative trial, because calcium at 1,200 milligrams and vitamin D at 800 IU are widely recommended in postmenopausal women. However, when vitamin K2 was added, the hypercalciuria and the hypercalcemia reversed. So we know that K2 is needed to ensure the delivery of calcium into the bones instead of the joints, arteries, and kidneys. And this is some of the most exciting information to come out of the recent research is on this K2. It's different than K1, and we're going to go into the details about the differences between K1 and K2 here and how we got so deficient in K2. It's a really interesting story, so stay tuned for that. But preview of coming attractions, the K2, the cool stuff that it does is it tells the calcification in the body where to go. So it pulls it out of the arteries and joints and kidneys, where we could have kidney stones or arterial sclerosis or calcification of the joints, and pulls it into the bone where we need it. And so I like to think of it as the guy on the tarmac at the airport with the little batons trying to tell the airplane where to go and which direction to go. So there's K2 in the bloodstream going, no, 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 get out of the arteries, get out of the joints, get out of the kidneys, and go right into the bone this way. So it's pretty awesome stuff, and it does so much more. So just to put this into perspective of where we were with the last seminar, or the last webinar, and those of you who have taken the seminar, following down the methylation pathway, we're trying to create methyl groups, and they're created by B12 and folate, and getting the correct kinds for your genetics is very critical. So these methyl groups then have a large effect on the repair, detoxification, energy production, and hormonal balance of the body, and variants in the enzymes needed to produce methyl groups or distribute them or break them down can cause disruption of many of our bodily processes. And what do I mean by variants? I'm talking about genetic variants, or some people call them defects. I like to call them variants. It's just a little less negative. There are changes in our DNA that don't allow us to make these enzymes efficiently. So lack of these particular enzymes needed to produce methyl groups can disrupt many of our bodily processes. And that's what we've been talking about with the methylation pathway seminars. And so many of you are coming to that. Maybe you're listening today. And again, this VDR I consider to be one of the most important pieces of this methylation pathway, because if you overlook this, then you're going to have chronic infections, your hormones are not going to work correctly, your immune system is not going to work correctly. So many things are going to go wrong. So addressing the vitamin D receptor, or the VDR, is critical, and not a lot of people are addressing it. When I look at the MTHFR support reports, they're not talking about it big time, and nobody's really talking about it. Amy Yasko talks about it a little bit, but not to the extent that I think it needs to be addressed. So again, a review. Now, DNA makes RNA, makes protein, and makes our enzymes. The DNA is the blueprint for making proteins of all kinds, and enzymes are the protein structures that speed up reactions in the body. So without our enzymes, biochemical pathways will be too slow to sustain life. So when we look at the vitamin D receptor, and let's say we have a genetic variant, there are three main genotypes in the vitamin D receptor, three main ones that have been studied extensively. There are actually many more. But the three ones that we have seen a lot of information coming out, a lot of studies being done, have a very real impact on the ability to absorb vitamin D. So if you've got a homozygous, or both genes, you get one gene from your mom, one gene from your dad. If you have a homozygous variant in any of those vitamin D receptors, then your ability to absorb vitamin D is going to be greatly diminished. If it is homozygous, you're reduced to about 30%, or reduced by 70% in your ability to absorb vitamin D. That's big. If you're heterozygous, then you're reduced by about 30% in your ability to absorb vitamin D. But even that is big. So you're still going to get a little bit. Obviously, you're still alive. If that gene didn't work at all, or that enzyme was completely absent, then it would be, you probably wouldn't be around. So again, review SNPs. These are single nucleotide polymorphisms, and they're changes in the DNA that may code for an enzyme needed for the body to function correctly. So changes in the DNA will alter the body's ability to make the methyl groups needed to sustain life. And genetic changes in receptors also have an effect on how the body functions. So we haven't talked so much about receptors before, more about the enzymes that we need to create methyl groups. So we also have to think of, okay, we can create a methyl group, but is your body picking it up? Or we're creating a hormone, but are your body's receptors actually pulling it in? And that's a whole nother subject. So this VDR, the vitamin D receptor, this gene encodes the nuclear hormone receptor for vitamin D3. It's also a receptor for bile acids, or lithocholic acid. And I'll talk about that in a minute, but it's involved in mineral metabolism, immune response. Also, mutations in this gene are also associated with type 2 vitamin D resistant rickets. Glucocorticoids are known to decrease expression of the VDR, so those people that are on steroids, it can interfere with the intestinal transport of calcium, iron, and other minerals. Other things can also interfere with the expression of that vitamin D receptor, which is fascinating. Just a preview of coming attractions, bacteria, viruses, Lyme disease can also take out the expression of the vitamin D receptor, which is pretty amazing when you think about it, that these guys are smart enough to take out our immune system through the vitamin D receptor. So fascinating. They've been around a lot longer than we have, and they're pretty smart. So we have ways to outsmart them. So back to our methylation chart. When we look at the two main circles here in this pathway, you can see this one here is basically our B12 pathway, and this is our folic acid pathway here. These two are our major generators of the methyl groups that we need to sustain life, to help detox the body, to help repair the body, to help repair DNA, to help make our hormones, our neurotransmitters, make energy, etc. So we kind of talked a little bit last time about, so all of the areas in purple, those are all possible genetic variants, or our inability to make those enzymes correctly will interfere with correct functioning of this entire pathway. But take a look right smack dab in the middle here. This vitamin D receptor is right here, and it's a big player. And we can see that it's affected by the methyl groups coming off of the generators up here. So these methyl groups actually activate the vitamin D receptor, and in turn it's going to make dopamine over here. Actually what it does is it increases this particular enzyme right here called the tyrosine hydroxylase enzyme, which converts tyrosine into dopamine, and that's how it actually increases dopamine. It's also a potent stimulator of the nitric oxide synthase gene over here, so it makes nitric oxide, which actually has a tremendous effect on dilating the blood vessels and getting oxygen to the body. And so many other effects, as we'll see as we go along, but I wanted you to get that into perspective of where we are here. So we've talked about the MTHFR. This is the gene that keeps us from activating folic acid to the active form 5-methyltetrahydrofolate. And we also had talked a little bit about these genes up here where we activate or recycle B12. So variants in these areas can keep us from making methyl groups in the first place. So that's where we want to definitely address those areas. But you've really got to address this VDR before you even go up here and address these areas. So that's the different thing that we've been doing in the seminars, is teaching you how each of these receptors kind of affects the other, and you can't just adjust them individually. You know, I see questions on chat rooms like, oh, I have the MTHFR variant. What do I take? No, it's not that simple. What else do you have? What are the variants do you have, and what's going on in the rest of the body? So you got to look at all of them kind of in a nice little, I call it a symphony. We got to look at all of them together. You can't just address one variant without addressing the others. So back to a lithocholic acid, or LCA. It's a bile acid which helps absorb fats. So the vitamin D receptor helps us in our fat absorption. It's made from bacterial action in the colon. It's what we call a secondary bile acid. The main function of bile acids is to facilitate digestion and absorption of dietary fat, but they're increasingly being shown to have hormonal actions throughout the body as well. Research suggests that LCA kills neuroblastoma cells and is cytotoxic to numerous other malignant cell types. That's kind of impressive. So, and fiber binds to the LCA to help with the excretion. LCA activates the vitamin D receptor, and vice versa. So, super important piece of the puzzle for helping our immune system function correctly and our absorption of fats. So here the vitamin D receptors are spread throughout the body. They're in the parathyroid glands, they're in the kidneys, they're in the intestines, they're in the bones. They're in the immune system, the skin, the bone marrow, reproductive organs, and pancreas. Actually, they're in small amounts pretty much throughout the entire body, but those are the areas where we have the most receptors, and you'll see why as we go on. So here's the vitamin D receptor sitting right here, and it's involved in gene transcription, protein function, anti-tumoral effects down in this. These are some of the things that it does: the immunological responses, cell cycle arrest, differentiation, anti-angiogenesis, and apoptosis effects in the body. It's sometimes thought that liver and kidney are the only sites of conversion of 25D into that 125D, or from the inactive to the active, but there's evidence that this process happens outside those organs, not coincidentally at the very sites where patients report symptoms of chronic disease. This is fascinating. You don't hear this so much. In sarcoids, little cells inside a person with sarcoidosis actually can convert 25D into 125D. Synovial fluid, like in rheumatoid arthritis patients, there's conversion happening there. In the macrophages of immune cells of the body, respiratory epithelial cells, so in cases of asthma, etc., you'll see conversion of 25D into 125D. It's just kind of fascinating. These disease site-specific peaks in 125D somewhat undercuts the validity of the that should say 25D serum blood test as the gold standard for measuring the presence of chronic disease. That's generally in the blood we measure the 25D, and I'll show you why in a minute here. So these are the three genotypes where we can find common gene polymorphisms: the APA1, the BSM, also known as the FOK, and the TAC1 genes. The most common genotypes observed in our population were a combination of those. So we have heterozygous A, heterozygous B, heterozygous T is in 37% of our population, a homozygous A, homozygous B, and a normal T would be in 20%, and etc. So you get the idea. And the most rare type is this homozygous A, heterozygous B, heterozygous T. It's in 9% of our population. So fairly common in some of the variants, and then not so common in some of the other variants. But again, the vitamin D receptor plus 125 dihydroxy D3 affects skeletal health, immune regulation, hormonal secretion, skin and hair. An interesting study that I saw on alopecia and how variants in the vitamin D receptor can actually lead to damage of the hair follicles and lead to alopecia. Central nervous system effects and calcium transport we're well aware of. Also, those low catecholamines come from low expression of the vitamin D receptor. Those with deficient dopamine levels may show signs of depression, low self-esteem, lack of libido, feel mentally fatigued, easily gain weight, or have a history of ADD, ADHD. Low adrenaline and noradrenaline will show signs of fatigue and depression as well. So how many of us are treating depression or low adrenal function on a consistent basis? A show of hands here, I'm betting everybody in the audience is saying, yeah, that's the most common thing I see is low adrenal function. And it can definitely come from low expression of the vitamin D receptor. We're finding, and in myself as well, I was one of those on chronic adrenal support and chronic immune support because I had chronic fatigue syndrome through Epstein-Barr virus activation. And this kind of depression, this sort of like, it's usually a kind of an apathy, weight gain, mental fog, low self-esteem, definitely, the depression, lack of libido for sure. And this all comes, and I do have a homozygous variant in the the TAC receptor, and half of one in the BSM receptor as well. So I always wondered, now, why do I have to take adrenaline all the time? And maybe it's just the stress I'm under or something. I couldn't figure it out, but I was happy that we had some natural remedies for, staying on top of the fatigue or all of these other symptoms. And as I began to address my vitamin D receptor issue, I don't have to do this anymore. I haven't taken adrenal support in close to about 1.5 years now, and that was unheard of. And I haven't had the depression just went away almost immediately when I started to address my methylation pathways and specifically my vitamin D receptor issues. When we look at the vitamin D receptor and bone health, calcitriol, the active hormonal form of vitamin D, acts through the vitamin D receptor to induce the synthesis of osteocalcin. That's the most abundant protein in bone. In studies of twins, variation in serum osteocalcin levels was shown to have a major genetic component. That was a study done by Kelly et al. 1991. And looking back, this was in 1991 that we were already aware of genetic variants in the vitamin D receptor, and that's why it's one of the most widely studied variants out there. And if you look on the SNPpedia is a great place to go if you're wanting to look at the best studies out there, the peer-reviewed journals, etc. So back in 1987 and 1991, we were already looking at the genetic diversity and bone density correlation with the vitamin D receptor. And so Morrison et al. In 1992 presenting evidence suggesting VDR polymorphisms may influence serum levels of osteocalcin. A lot more studies being done, and they're very current. I just wanted to point out that it has been going on for quite a while, studies of this variant. So when we look at chronic renal disease as well, it can affect the VDR activation, but I wanted to point out some of the other effects here from abnormal mineral homeostasis, increased inflammation, increased thrombogenesis, vascular dysfunction, and deregulation of atrial natriuretic peptide, etc., leading to increased cardiovascular-related mortality. It's fascinating. So the vitamin D receptor is a control system of sorts. I want you to get this, because this is a really interesting theory here. So when exposed to infection and damage, especially that which is caused by pathogens, the body begins to convert the inactive form of 25D into the active form 125D. So think about it. If you have an active infection in the body and you go in and you measure 25D and it's low, well, you think, well, I'm not taking enough vitamin D. I need to take a bunch more. What you're actually doing is increasing that conversion from 25D to the active form 125D. You're not going to measure it very accurately in the blood. Make sense? As cellular concentrations of 125D increase, 125D increases the vitamin D receptor, turning on any number of genes the receptor transcribes. Okay, so that's we get. So what happens? The vitamin D receptor then significantly affects 229 human genes in the body that we know of. Many of these genes are associated with immune dysfunction and free radical pathology, being associated with some of these diseases here as well. The activation of certain genes also leads to the synthesis of antimicrobial peptides. The antimicrobial peptides are the body's natural antibiotics and have a potent antibacterial effect. So if that vitamin D receptor is working correctly, it's taking that inactive form of vitamin D that's been activated and then turning on some killers, turning on our killer cells. Antibacterials, calicidin is one of them. It goes in and just kills these bugs pretty effectively. But imagine that you don't have a vitamin D receptor that's working well, or it's working at only 30% of its normal function or less, and the body can't take that active form, that 125D, and turn on these antibacterial effects. So VDR is also, like I mentioned earlier, inactivated by bacteria. So bacteria can inactivate the VDR and the innate immune response. This allows the microbes to proliferate. In response, the body increases the production of 125 from 25D, leading to one of the hallmarks of chronic inflammatory disease. And that is when we measure it in the blood, a low 25D and a high 125D. So you can see what's happening here, and you can measure it more thoroughly if you're looking at both of these markers. And that's just not done. I've never seen it done. I asked for my I went to LabCorp and asked them to do the 125D levels, and they just looked at me like I was insane. Like, no, we don't do that. We only do 25D. Why would you want to do 125D? They just looked at me like I had 3 years. So anyway, I think it's an important thing to look at in the future. I think we're going to be looking at both of these markers. So this pattern is a result of the disease process rather than a cause. So we look at, we're looking at 25D levels as a static number, that the more we take, the more we have sitting around, and the better off our immune system is, when it's just not that way at all. And one of the pioneers in this field is Trevor Marshall. If you get a chance to look at his website, it's really compelling. His research is quite compelling. He's a PhD, but he's focused on this, not necessarily the vitamin D receptor, but levels of vitamin D in the body and how we should measure both 125D and what happens in the inflammatory processes and in disease processes. So I encourage you to look at his research more. So when the immune system is challenged by pathogens, then the body activates this enzyme. This enzyme is called CYP27B1, and it's actually in the cytochrome P450 enzyme family of the liver detox pathways. And it causes more 25D to be converted to 125D, which of course increases the activity of the VDR. So if you've got a variant in this particular gene, which you can have, I looked it up and it's like, oh, dang, there's another gene where we could have some issue. If we don't make it correctly, then we can't convert 25D to the 125D. Okay, so there's another issue. However, just because the concentration of 125D reaches high levels, sometimes extremely high levels, does not mean that the hormone is successful in binding and activating all of the body's vitamin D receptors. In fact, a 125D that is elevated for an extended period of time suggests to us that the activity of the VDR is partially blocked. So that's one way that you can tell whether there's a vitamin D receptor issue here, aside from getting your genes tested and just getting some blood work done. If you see a really high 125D and a low 25D, then you can guess that that vitamin D receptor might be blocked. So when bacterial ligands block the vitamin D receptor, the receptor is prevented from transcribing another hormone, the CYP24A1. This is a well-studied enzyme which breaks down the excess 125D. That's the other issue is we don't want a ton of vitamin D running around, active vitamin D, overstimulating our immune system, because you know what happens when you overstimulate the immune system. And we call that an autoimmune disorder. So if we don't make this enzyme correctly, or the vitamin D, this vitamin D receptor may not work correctly, or the cytochrome P450-24A1 gene has a variant or isn't transcribed well because of a bacteria, then we've got high levels of 125D, which can then lead to immune dysfunction in the form of a possible autoimmune disorder. Makes sense. So again, here's cholecalciferol, vitamin D3 in the liver being converted to 25-hydroxy-D3, in the kidney being converted to 125-hydroxy-D3. Then the VDR is activated, and then we have the genomic and non-genomic effects in the body. But also here, this is the inactive form of vitamin D3, and this is the gene that does that, inactivate. So when it gets too high here, we're going to turn it off. All right, wait, we have too much. So we could have variants in this gene, or this gene that can lead to difficulties in making this whole pathway work correctly. So what happens when we have excess 125D levels? The following diseases and conditions manifest with high levels of 125D. When we looked it up, we saw all of these particular areas or diseases or conditions or imbalances, however you want to say them, associated in the literature with high levels of 125D when it was measured. Hashimoto's and autoimmune disorders are associated with vitamin D in the literature. Vitamin D dysregulation, these were a study. This was a study out of the Annals of New York Academy of Sciences, 2009. The vitamin D dysregulation prevents the breakdown of the active vitamin D metabolite, 125-hydroxyvitamin D. And when 125D rises above its normal range, it binds the alpha-beta thyroid receptors, or the glucocorticoid receptors, and the androgen receptors. And you can see how that can cause an array of hormonal imbalances. If we can't break down our glucocorticoids, our androgens, then we're going to have major issues. And if T3 is displaced from the alpha thyroid receptor, thyroiditis may result, and since the VDR, GCR, and AR also express multiple families of immune molecules, expression of these natural antibiotics further declines in response to dysregulation by 125D. The end result is a system-wide drop in the immune system that may allow pathogens to spread with greater ease. And we found, you know, I've mentioned before that we have had, you know, some correlation in my practice. I've seen Hashimoto's. I have 6 cases where the thyroid peroxidase enzyme was able to return back to normal levels when we started addressing methylation pathways and specifically this vitamin D receptor. So it was pretty amazing to see when we started working with this that Hashimoto's would start to respond to this. And I haven't seen a system-wide study on that, but it's I think something going on there. So that's my theory. Vitamin D receptor and autoimmune dysfunction. Autoimmune diseases are caused by infections. This is the theory here. These infections are able to persist due to the inability to fight them due to the inactivation of the VDR gene, either genetically or by the infection itself. There was a study done by Perez showing that 81% of a group of 54 patients representing 20 different autoimmune diagnoses reported continual improvement after treatment durations of 18 to 53 months. He used a VDR agonist, it was a drug, I guess, and antibiotics, further pointing to bacteria as a causative agent in autoimmune disease. So he was having an 81% success by using an agonist, something to increase the function of the vitamin D receptor, and also at the same time going after the bacteria with antibacterials. I think we can do the same thing in a more natural way, but I thought it was an impressive study. So here's the new Bloodwork Suggestions. Scientists and clinicians should be encouraged to test both 25D and 125D in their subjects. A low 25D and a high 125D are both useful markers of the disease process. Low levels of 125D are more likely a result rather than a cause of disease progression. And dysregulation of the VDR by chronic intrahepatocytic microbiota, the bad bugs, would cause significant hormonal disruption by allowing 125D to accumulate and displace the negative ligands from alpha thyroid, glucocorticoid, androgen, and other nuclear receptors. So by reducing the ability of these same receptors to express the antimicrobial proteins, essentially, accumulating 125D would also cause a system-wide drop in the expression of those antimicrobials, allowing pathogens to proliferate. So that's out of the Annals of New York Academy of Sciences. Very interesting study if you get to go take a look at that. So when I looked at conditions associated with the VDR genetic variants, I went to the SNPpedia and looked up the studies, and I found information on all of these conditions associated with genetic variants in this receptor. So you can see that it covers a lot of ground. It's big. So when we look at, again, the vitamin D blood work and why they only do the active, here's the reason. So the half-life of vitamin D is around 24 hours, thus making the concentration of serum dependent on the most recent exposure to sunlight and vitamin D ingestion. Similarly, the half-life of 125-hydroxy-D is 4 hours, and its production is tightly regulated by the calcium needs of humans. So it's going to vary. Modern medicine believes that due to these reasons, there's little usefulness in measuring vitamin D or 125D in the circulation. So they use the serum levels of 25-hydroxy-D, or the inactive form, because it stays around for about 3 weeks. This rather long serum half-life serves as an accurate indication of vitamin D stores obtained from both ultraviolet radiation and dietary intake over long periods. So that's their reasoning, is that it's, it sticks around a long time, so they figure it's a good way to look at what's going on in the body. After looking at the research, I'm questioning that. So what are our levels that we like? I found, again, different levels that they thought were good. The Vitamin D Council says sufficient is 40 to 80. Endocrine Society says 30 to 100. Food and Nutrition Board, says greater than 20 is sufficient. And some other testing laboratories will have a range of 32 to 100. And toxic, they say, is 150. But as we saw, as we've been talking, it's not a real good indication of what's going on in the body. The Vitamin D Council suggests that a level of 50 is ideal level to aim for. This is why the Council recommends that adults take 5,000 IU a day of vitamin D supplements in order to reach and stay at this level. And I have to say that is so short-sighted. And maybe you guys, I'm a muscle tester, I'm a kinesiologist, and for the longest time, I knew about, low levels of vitamin D being a problem, seeing low levels in the blood work on my patients, but why would it not test good? I didn't understand it. I put vitamin D on people's bodies and it actually tested adverse. What is going on here? They have low levels. Why aren't they absorbing it? And I'd seen in references, oh, you have to take it with the biggest meal of the day, or the biggest fat-absorbing meal of the day to get it to test good, or you have to take it with some kind of gallbladder support. Maybe it indicates low gallbladder function. And as I began to study this methylation, I'm like, oh, I see what's going on now. So either they've got a vitamin D receptor issue, or they need other cofactors in order to get that vitamin D activated, and that's where we're going next. So I'm going to jump past this one for a minute. So don't forget, we have the structural difference between vitamin D2 and vitamin D3 is the side chain of D2 contains a double bond between carbons 23 and 23 and that little methyl group on carbon 24. So we know that vitamin D2, if we get it in our diet, has to be methylated before it's turned into D3. Methylation is involved in the entire activation of the D3 molecule all the way through. So if we don't have the MTHFR is not working, or the MTRR is not working, or we have variants in those genes, then we know we're not making methyl groups and we can't activate that vitamin D. So you can see how important that is in the whole process. I'm going to go, we already covered this, so I'm going to go forward. Alrighty, and we've already covered this as well, so I'm going to go forward here. So the three types of D that we carry at, let's spell that immuno wrong, I think. So at NutriWest are the complete high D3, and this is a 5,000 IU. I use quite a bit of this. It's from a cod liver oil source, or vitamin D 400, which is, again, from the fish liver oil at 400 IU. And then the Immuno D3, which has some added things in it, quercetin, beta-cytosterol, lactobacillus, acidophilus kind of units in there that help with the absorption in the intestines. Some options. But what I want to point out is the importance of vitamin A, and this is where we have to look again at the cofactors here, vitamin A and vitamin K, and how important they are in the absorption of vitamin D. So retinoids, there's a retinoid X receptor that is part of the vitamin D receptor. They're called dimers. It's right in there with it. So the retinoids are metabolites of retinol or vitamin A, and the retinoid acceptors are receptors for vitamin A. So the RXRs are also cofactors required for activating thyroid hormone and vitamin D. And vitamin A signals play critical roles during embryonic development. In particular, heart morphogenesis depends on vitamin A signals mediated by the retinoid X receptor, as the systemic mutation of this receptor results in thinning of the myocardium and embryonic lethality. So it's important. So there it is right there. There's that party that I talked about with all the curling ribbon. But here's the part of it is a retinoid receptor or vitamin A receptor, and there's the vitamin D receptor. So you can't ignore the vitamin A there. The whole system will not function correctly without it. Vitamin A and vitamin D work together to help your genetic code work properly. If you don't have enough vitamin A, vitamin D may not be able to perform this function properly. On the other hand, research shows that too much vitamin A and vitamin D does not work as well. So it's getting the right amount in there, and that's why I love the muscle testing, and if we can get it in the diet, that's great. So the vitamin A emulsion, each drop contains 5,000 IU and a little bit of vitamin E in there as well. Vitamin K, let's not forget about vitamin K. When I started looking into vitamin K, I was blown away by all the things it does. It's just it's a big topic, and it's one of the most exciting areas of research coming out now, and it's very new. In fact, since 2009 is really when things started to happen for vitamin K research, especially vitamin K2. Vitamin K1 we find in our diet. It's in our green leafy vegetables, and vitamin K2 is made by bacterial fermentation. I had a theory about why don't we make vitamin K2 anymore, and why don't we have it in our diet? And you've probably heard me say this at seminars that our bacteria in our intestines is much changed from where we were, where our ancestors were, and our ability to have good flora in there that converts K1 and our green leafies to K2. I think that's a piece of it. I think our intestinal flora has definitely been damaged by various things, the hormones, estrogens, by growth hormones, by genetically modified foods, etc. And definitely our intestinal flora is not the same. But there's more to this story that I just learned recently, and it's just kind of fascinating. So K1 is mainly our blood clotting factor. K2 is more involved in strong bones and teeth, and it's the one I told you that kind of directs calcium where to go, in fact, into the bones and teeth. It may help protect against developing conditions like cardiovascular issues, free radical damage, and brain degeneration. And vitamin K is important in making sure that calcium you get from foods and supplements is used in your bones. So some of this research came from Weston Price, and I think some of you know Weston Price's research on nutrition and how nutrition affected. He was a dentist in the 1930s. He began to do research into why modern man had so many dental cavities and changes in our dental arch and teeth that were crammed together, all kinds of issues in that area, and why other societies did not. The people that were consuming a traditional diet, he found, were not having these dental issues, and he began to look at what were they consuming that we weren't consuming, and he found something he called the factor X. And he knew it was in some fermented foods. They were eating, fermented cheeses, fermented butter, and a lot of their food was fermented because they didn't have refrigeration and had to preserve their food somehow. But then again, there's a missing piece there that I just kind of discovered through reading a book called The Calcium Paradox, Vitamin K and the Calcium Paradox. Incredible book. It's really recommended. But she says that if you aren't consuming grass-fed beef, which our ancestors were, grass-fed beef, if you think about it, the cows are out there and you're eating a lot of grass, which contains high levels of vitamin K1, and in their intestines they're converting it to K2, and this is where in our diet we were getting a lot of K2. So we had butter and cheese and the meat from these animals when they were grass-fed. So when we started to introduce agricultural methods of raising cattle and other game or other food sources, chicken, etc., we began to feed them grain, and they didn't get their K1. There's not any K1 in grains. It's in our green leafies. Our levels of K2 began to just really decline, and that was really the advent of the cardiovascular issues, cancers. I'm sorry, I'm not allowed to say that word. So free radical pathologies and other issues along the way, our chronic diseases of man, were beginning to show up right around that same time. So that was a fascinating piece of the puzzle. Sometimes you just have to tell calcium where to go, and that's what vitamin K does. High vitamin K2 status was associated with lower concentrations of inflammatory markers. So inflammation, we know, is involved in every chronic disease in the body. This is out of the American Journal of Epidemiology, 2008. Low intakes of vitamin K2 may increase the risk of hip fracture in women. This is the American Journal of Clinical Nutrition. K2 and its derivatives have a favorable effect on programmed cell death and enhance the effect of all trans-retinoic acid. That's vitamin A. That's out of the European PubMed Central. Vitamin K2, and this is called menaquinone. This is a menaquinone form of K, continues to shine as a vitamin that supports multiple aspects of bone health. K2 transports calcium from circulation into bone tissue by activating carboxylation of two key calcium transport proteins in the bloodstream. And those two proteins are the matrix GLA protein, and that transports calcium from circulation into the bone. So that's out of the arteries, out of the joints, out of the kidneys. And the other one is osteocalcin, which reduces calcification of the arteries and plays a significant role in bone development. Both of these are going to pull calcification out of the bad areas and put them into the good areas. And this was the interesting thing, is that Weston Price was starting to see cavities disappear when he began to give these folks the active form of vitamin K or K2 in the diet. He began to give them these fermented foods, and they started to reverse cavities. It's like, whoa, that's kind of cool. He didn't know what it was called. He didn't call it, it wasn't called K2 until much later, but at the time he was calling it factor X. He didn't know what was in there, but he knew something miraculous was going on. And so we're seeing the same thing happen with our arteries and bone. So it's very exciting. So again, the matrix GLA protein, it's an inhibitor of vascular calcification, and the osteocalcin promotes bone mineralization. That's the menaquinones. This was kind of interesting, and this came out of that vitamin K and the calcium paradox. Studies show that adding menaquinone to the diet will activate MGP to reduce arterial calcium content by 50% in over just a 6-week period. I was blown away when I saw that. Are you kidding me? Like, we reduced arterial calcification by 50% over a 6-week period? Why isn't everybody taking K2? I don't know. Because we're certainly not getting it in our diet unless you're going out and you're getting grass-fed beef, grass-fed butter, grass-fed cheese. Get grass-fed everything you can get, because that's how you get your good K2. And natto is the other form. That there's two forms, K4, K7. K24, K27. And the K27 is mostly found, it's the more potent form, mostly found in a fermented soy product called natto, which I hear is like super nasty. This author was saying she tried to like it. She just really couldn't do it. She tried it in all different ways and tried disguising it, but it's just gooey, sticky, mucusy, stinky. She said it smells like stinky socks. I haven't been brave enough to even go there, so I'd rather swallow a pill. Sorry, but I would. And go pick up some grass-fed beef. And this is one of the best reasons for doing grass-fed beef. You know about the benefits of the good essential fatty acids, but who knew about the K2 in there? I sure didn't. So again, these are the two forms, K24, K27. Research continues to validate the vitamin K2's benefit to bone health and heart health. Two common supplemental forms of K2 are the menaquinone 4 and menaquinone 7, with the number designations referring to the length of the molecular side chains. There's another study that was done in postmenopausal women with osteoporosis. Just vitamin K supplementation achieved significant improvement in vertebral bone mineral density and reduction in overall risk factors. And I had one doctor who said that he saw vitamin D levels he was not able to get up, calcium levels were leaking out of the urine, and he did nothing but vitamin K2 and found both of those reversed. So vitamin D levels came up, and the loss when he measured it in NTO peptides in the urine stopped. The calcium loss stopped. So this was another really interesting study where menaquinones can help keep skin firm and healthy. This means wrinkles. Yay, those of us who are looking at getting older and we're looking in the mirror every day and going, oh man, I'm not happy about that. Actually, K2 can benefit in that area. Actually, we're seeing studies showing that it increases vascular health of leg veins or varicosities, and activation of osteocalcin has a significant impact on the body's production and sensitivity to insulin. So that's insulin receptors. So our blood sugar balance can be beneficially impacted by K2. So again, where does this deficiency come from? Disrupted intestinal flora for sure. Lack of K1 and beta-carotene in the diet, yes indeed. Lack of grass-fed meats, big time, eggs and dairy products. Increase in consumption of processed foods and lack of fermented food in the diet, absolutely. So Complete K is a product I designed with a little bit higher levels of vitamin K4, K24, and then a little bit lower to K27. The reason being K24 has a shorter half-life. K27 stays around a lot longer, so we needed higher levels of K24 to get it going in the body. A little bit of K1 in there for clotting factors. So vitamin D3 and K2, these findings indicate that combined administration of D3 and K2 compared with calcium administration appears to be useful in increasing bone mineral density of the lumbar spine in postmenopausal women and with osteoporosis. And that's out of the Journal of Orthopedic Science, November 2000. Don't forget your other cofactors, calcium, magnesium, zinc, and boron. So the conclusion we can make here is that there's a strong reciprocity between the vitamin D system and epigenetic mechanisms. And we know the vitamin D system is on the one hand regulated by epigenetic mechanisms and on the other hand is involved in regulating epigenetic events. The vitamin D receptor might even regulate DNA methylation. Epigenetic regulation of gene expression is a fine-tuned mechanism, and its dysregulation can lead to pathological conditions. The impact of vitamin D on the maintenance of normal epigenetic landscape underlies the central role of this hormone in physiology, and regulating epigenetic events can be further mechanism by which 125D3 may prevent or delay tumorigenesis and onset of chronic disease. Therefore, we need to better understand the impact of vitamin D on the epigenome and plan thorough and comprehensive studies to examine this interplay. I didn't make that up. I didn't say that. That came out of the vitamin D article called Vitamin D in the Epigenome, Frontiers in Physiology, April 2014. That's how new a lot of this research is, but they're saying, all right, we need to look into this big time. I really think more studies need to be done because it's exciting and it's important. But the good news is we can supplement with vitamin K2, vitamin A and vitamin D, and bone-building minerals, along with supplementation of essential fatty acids, to begin the process of undoing years of deficiency. So it's not just preventing, it's undoing, which is very cool. I looked at my mom today. You know, I bring up my mom because she's cool. She's my guinea pig. She's 90, super strong, and I swear her wrinkles are getting better. I'm going to have to put, next webinar, I'm going to put a picture of her in there because I'm looking at her and going, dang, Mom. They think that she's around 70, 75, people that look at her, and so we're not even telling them. But she's as strong as it gets. She's on half a blood pressure medication, and even that's her blood pressure is going down with this methylation process. So that's super exciting for me personally. But this ultimately will be an incredible insurance program against all the possible outcomes that deficiency of vitamin D can lead to, including cardiovascular disease, free radical pathology, brain dysfunction, blood sugar imbalances, bone health, immune dysfunction, and more. And this is the true spirit of the science of epigenetics, the knowledge of the genetic defects that have held us back and doomed us to poor outcomes can finally be bypassed and modified through knowledgeable supplementation protocol. I really think this is a truly exciting time to be alive. I'm super excited about it. I'm glad you guys are too, because I think it's pretty cool. So to learn more, I really advise you to attend the methylation seminars. Nobody's doing this the way that we are, and combining the muscle testing with the methylation to really find out what's going on in the body. Go to the SNPpedia and study the VDR effects, and all of the studies that have been done are in there. And then get your genetics tested. We've previously been working with 23andMe, and we're a little bit unhappy with them. A couple reasons. One is they denied selling to chiropractors. According to Dr. Tui and some other doctors, they won't give us some kits to give to our patients. And they just doubled their price. They went from $99 to $199. I did find out that Ancestry.com will do the test. It's $89 right now. And you can get your raw data, and that's really all we want. We want the raw data to put through our genetic templates so we can pull out your vitamin D receptor and these other genes that we're looking at. Get your vitamin D levels and get them both tested, the 25D or 125D, see what's going on there. Go look at Amy Yasko's information. She's brilliant. So much information she's done, and she has a lot of great webinars on YouTube. And then go to geneticgenie.org to learn more. These are the methylation seminar dates and locations. Or you can talk to your NutriWest distributor. We are all over the country, and we're out. People are just so excited about it, and we're being asked to come back and do it again. It's kind of a complicated topic, but man, you can see how important it is, and it's worth studying and worth doing for those chronic patients. We're getting just tremendous results, and so hopefully we're going to be in your area sometime soon. Or you can come to one of the other seminars in other areas. But talk to your NutriWest distributor and see where we are. And then if you want to learn holistic kinesiology, if you don't know muscle testing, and I really advise you to learn it, we are doing a basic course in Phoenix starting in February in 2 parts, the basic and advanced parts. And then we'll be doing more of those around the country as we see if people are interested. But I know some people come to these seminars and they don't do muscle testing, and you can do it. It's just a lot of blood work, a lot of testing, and so much easier to see what's going on. If you just take your genetic test and do the muscle testing with it, you can find out really where these variants are being affected. So that's my story.
Thank you so much, Dr. Dunn. That was fascinating, and I know that we have a high interest from our audience. We did have one of the questions that they wanted to answer that you did go over most of it, by the way. Thank you. Once you get the raw data, can you go over where it gets plugged in?
Yes, thank you for that question. The 2 main websites that we've been using are the Genetic Genie, and that one is free. You just upload your data in there, and you can get that's the main methylation variants you'll find in there. It'll give you the VDRs, the 2 VDRs, 2 of the main ones that we're looking at, not the 3rd one, but it'll give you the TAC variant and the BSN variant, and then a bunch of the other methylation variants. But also the MTHFR support website, which is $30. You can upload the raw data there, and it's going to give you 42 pages of variants that are very fascinating. Again, they don't pay a lot of attention to the VDR, and I think they're doing a disservice there. I'm putting it out there that I would very much like somebody to help me create a template. I don't know what I'm doing. I don't know how to program anything, but I would love to make our own. Even if somebody at NutriWest, if we could find somebody to make a template for us so we can get the exact genes that we are interested in.
Yeah, I think that judging from the questions, this is going to be something that we might want to address head on here, because several people are saying that geneticgenie.org and ancestry.com, they're just not containing the data that 23andMe does. So I think that maybe one of the things we need to consider is contacting 23andMe to be more amenable to working with our group of people, because we have the capacity to refer a lot of people to the best organization. We just need to make sure that they, will work with us.
Right. And, well, the nice news is that with Ancestry.com, you can download your raw data. So we don't even care about 23andMe. If Ancestry is going to play with us, we can get it for $89 right now. And that sounds good to me, because they've gone to $199.
Right. So price is an issue, and someone is saying that the raw data was less at Ancestry.com. So I guess we just have to, address all the issues and then get back to everyone on what we can do. And there's someone else that says, okay, we've got a lot of feedback for this for the webinar today. This is great. Someone said you can make your own templates at looks like livewello.com. I can't.
Oh, I did. I just don't trust their data. I have my own template there. The Dunn methylation template. I've made it. It gives me exactly what I want, but I have had some issues with Livewello, because they'll say, oh yeah, you have the VDR. MTHFR support will say, or Genetic Genius will say, yeah, you have the VDR, but they say, no, you don't. So there's some discrepancies. I'm not really trusting their data.
Okay. Well, yeah, and the comments on the different templates are still pouring in. So I think that we're going to have to end today's webinar, but we will keep tossing that around and keeping you posted as to the best and most efficient way to approach this, and we'll see what we can do in the way of templates and whatnot. But Dr. Dunn, could you give everyone an idea of what you will be presenting at the Winter Symposium in San Diego, which is going to be awesome in January? And I know I will be there. My husband, immunologist Dr. Belgrau, will be there, and I know you will be there. Dr. Murphy will be there. Dr. Randon Lundell will be there. It's just going to be a great combination of, a get-together of meeting of the minds. And maybe you could just give us a little bit of insight as to what you're going to be going over.
Yeah, thank you for saying that, because it's really impressive, the group of lecturers that NutriWest has put together. I got to say, there's some amazing talent there, including yourself, who I've always admired your work. But the quality of speakers there is just incredible. But I'll be talking about methylation. I don't have the whole weekend to cover it, and it's a big topic. So we will be covering the basics for those people who haven't had an introduction to methylation. We'll be doing that. And then Thursday night we'll be testing people. I'm bringing a few practitioners down, and we'll be doing some actual hands-on testing with people. So if you want to come in and watch and see how that's all put together, but I will be demonstrating that as well during the seminar, how to put it together with muscle testing, and then information on epigenetics and how to modify the genomes.
Wonderful. Great, and that the symposium does start Thursday, January 21st, and for anyone who is interested, you can call your NutriWest distributor or the NutriWest home office at 1-800-443-3333. So 2 4s, 4 3s. Again, that's 1-800-443-3333. If you're not sure who your distributor is, we'll get you that information so that you can come join us in San Diego for a great venue for a really great seminar. And Dr. Dunn, I'd just like to thank you for shedding light on a fascinating subject, and this is really great. As I said, I can tell by the response from people that there's a lot of interest and a lot of questions about this, and we're going to try to do some research and get some answers there. So thank you again, and listeners, thank you again for listening in. Remember that the webinar will be posted on the NutriWest website within about a week. So thanks again, and please join us for the next one. Bye now.